from Sri Chitra Tirunal Institute itself. He has more than 20 years professional experience in the field of evaluating biomaterials and medical devices as per international standard. As a study pathologist in preclinical research, he has evaluated a wide range of devices including heart valves, vascular grafts, coronary stents, bone and dental implants, etc. etc. He has authored more than 60 international publications and he presented his findings in many national and international conferences, also authored many books also. Today he will be delivering a talk on biological evaluation of biomaterial or medical devices. This topic is highly relevant as safety and biocompatibility of critical aspects in the development of clinical and translation research. Now I invite Dr. Sabrishwaran to deliver his talk. Welcome you, sir.
Thank you, Dr. Sibi. Respected principal and the organizing team, I personally thank Ms. Asha and Vipin for inviting me for this thing, and I congratulate as well as my best wishes for the the occasion of Dr. Sandhya, Professor Sandhya, Madam, and her retirement, and I am really thankful for inviting me to give a lecture on this day. So thank you, ma'am. Also, my wishes to the students and the audience, the distinguished faculties present in this audience group, and my colleagues. So without taking much time, I will go to the talk. So this talk is about the biological evaluation of biomaterials and medical devices, and the introduction was given by Dr. Vipin. So without much time, I get into this topic. Probably many of the students at the graduate level may not have much exposure to this medical devices. Anyway, I start with a couple of slides on the introduction, then the experience which we gained in some of the medical devices which we used, and on that or over and above that, if you have any clarification, you can stop me in that time itself and we can discuss about some of the R&D opportunities in that thing. So that's how we can plan this lecture. So, as I working in the biological evaluation of medical devices, it is applied. Many of us might not have much experience in the medical devices area, but we have seen in some of the hospital setups, some of your relatives or some other areas, you might have seen some of these names of some of the medical devices. For example, artificial heart valve, coronary stents, then knee joints, artificial knee joints. So these are nothing but the synthetic materials. Okay, either it can be a metal or it can be a polymer. Even syringes, needles, all those comes under the category of medical devices only. So naturally we will be knowing that our body, means we classify the science into biology and a non-biology substance. So biology means whichever living in this universe or in this world which is considered as biological things, and non-living things we consider as engineering goods or something else. So how they are going to help us for the biological system. So that's what we see in this biomedical devices. So it is mostly interdisciplinary. It is not pertaining to a particular department. We cannot squeeze into any one of these things. It is interdisciplinary. So here anybody, any person with science background or non-science background also can contribute in this thing. So with that aspect only I am giving this talk to you. So in many areas we use these medical devices. Some of the things for the understanding I listed here, but the domain is very large. More than 10,000 super specialties are available. So in that they are using these medical devices. So for the better understanding, cardiovascular area we use medical devices, orthopedic area, muscle, neural, dental and eye, ear. So all those areas we use medical devices. I'll give you some of the medical devices. So this everyone will be aware of this medical device, that is intraocular lens. For cataract surgery, many of the after 50s they use this synthetic thing, and this is one of the well known and very having a long history in the development of medical devices. So this is again a paradoxical innovation, we call it as, or a serendipity also we can call that it is an innovation. So this is after the Second World War, the usage of this intraocular lens was predominant in the medical field, because again the shattered glasses of the aircraft which was inside the pilot's eye, which later when they examined, they understood that this material was non-reactive. So they understood that it has some potential. So they used, because the glass is a transparent thing, and inside our eyes also we have lens which is transparent and causes it helps us to pass the light. So that's how vision we are getting. So based on these small clues. So here none of the other so-called biological scientists is involved. Only the aerospace engineering they were, and other pathology people they have involved in this thing, in the innovation of this particular medical device, and they started utilizing this as a intraocular lens. So this is one of the oldest medical device, well known innovated things. So it has a central position and it has a anchoring position. It will be placed inside our eyes in the place of lens. So that's how it functions as a medical devices. Then the next, cardiovascular system. So this is a blood vessel. Inside this, it's a pictorial representation. So inside a small blood vessel you have some mesh like thing, on the other side you have the real dimension of the metal mesh. So most of the time we might have heard about angina pectoris, that is heart burning or heart ailments. So where you have the heart attack, or in the common terminology we call it as heart attack. So in that, nothing but the condition in which the blood supply to the cardiac muscle, that is the pumping, our heart is the pumping mechanism inside our body, which is affected. To alleviate that, adequate blood supply is required. So the blood is given by the blood vessels, and if there is obstruction is there, the blood flow get affected. So naturally the oxygenation of our heart muscles get deprived. So that's how the problem occurs. So that can be revived or addressed. So this is how this particular stent, coronary stent, is being used. This is nothing but a metal mesh. So it is initially it is stainless steel, then later they made new alloys. So here the metallurgists, they have helped to find out this so-called, many other aspects are there that maybe you can Google it and you will find it out, why this particular metal is selected, not other metal is selected, that and all you can find it out. And the bottom two things is nothing but, that is the artificial blood vessel. So the later, once the science progresses, they found that we can even replace the blood vessels. So blood vessel is nothing but a elastic recoiling structure in our body. If there is 10 ml of blood, it will hold. If there is a 20 ml of blood, it will hold, because it will expand. So such a nature is the blood vessels condition. So similar things, we can, this is nothing but our fabric only. This is polyterephthalate PET, they call it as PET, polyethylene terephthalate. So polyethylene terephthalate, we have many examples. I do not find a PET bottle here. So that is also a polyethylene terephthalate, that is a fiber. So if we make that polymer into a fabric, we can create this blood vessel. So that is what you have seen in the bottom picture. So that is nothing but the Mahatamani, that is our major blood vessel. So that's what here being simulated. So in that way also this will be useful. So this is again just magnified images of the metal stents. So this deployment and other aspects I am not going into too much into this thing. So that is the medical domain. And this is the next one. So again the science progressed further, and they have even the replacements of valves. So our heart has 4 valves. Between the auricle and ventricle 2 valves are there, mitral valve and tricuspid valve, and in the aorta there is 1 valve is there, and in the pulmonary artery 1 valve is there. So the main function is this function as a one-way gate. It will allow the flow of fluid from one side, from A to B only, not B to A. If B to A flow is there, that is called heart failure. So in such condition, in the native, natural heart, if such is the condition, we call them as a cardiac patient or heart failure patient. So So that can be addressed by replacing these valves. So that is also can be done by non-living things. So these are the different models of heart valves. The top row is the mechanical heart valves, where it is a mostly made up of polymer. That is, the central portion is called a disc, and the surrounding portion is called a ring. So this will be placed inside the heart valve region, and this will allow the one-way flow of body fluids. Okay, so the that A and B are tilting disc model, and C is the ball and cage and ball valves. So here also the physics principle is mainly used. So one is the C1 is the ball and cage model. So that is based on the buoyancy law. That is, a polymer, a plastic lightweight substance float in the fluid. So in the ventricle chamber, when the blood loaded, naturally the ball will float and it will occlude the lumen, so that it will not allow the ventricle to auricle blood flow. Only from auricle to ventricle blood flow only will be allowed. That is called a normal cardiac cycle. So that is again, so the innovation is from the physics side. The next one is the tilting disc. A and B is the tilting disc model. So here again it is a lightweight plastic. So it will close and open in such a way, they design it such a way that it will close in an angled manner. So again there is a engineering innovation. So in how much angle, if you position that disc, it will properly close. So this is, and the bottom is nothing but your biological valves. So further, so and the hatch, that is the last and the corner. So that is the latest innovation in the cardiac valves. So A, B, C, D, E, F, G, all the E, F up to F, you need open heart surgery. So you have to open the chest cavity and you have to stitch the valve inside the body. So that is the requirement of this thing. But in case of G and H, now there is called a new domain in the interventional cardiology. So anything from remote outside your body, you can reach any part. Wherever the blood vessel goes, you can reach. So that is the medical innovation. That is the advancement which has happened in the 21st century, in the beginning of the 21st century, we can say. So the G and H and all, nothing but either through your hands or through the thigh region, you can insert this in a squeezed manner and you can place it in the heart. So that much the medical, due to imaging and many other, that's what it's a interdisciplinary. Not only a single person achieve this kind of things, only a team can achieve this kind of thing. So that is the latest one in this scenario ATWALS. So all these things involves innovation, evaluation at every point of time. So I'll just touch upon in my talk about the biological aspects of these evaluations. Yeah, this is the transaortic valve replacement, that is transcutaneous valve replacement, that is called a TAVR procedure. Even if you Google it, you will find it out, even YouTubes and everything, how they are doing it. So this is how inside the heart, from the remote area, they will go and place this valve inside the things without doing a surgery. So that what is the advantage is that, so instead of staying for the hospital for 10 to 15 days or more, we just stay for 3 days or 2 days. So that's what the lifestyle is being improvised. So that is the advantage of these kind of innovations. And subsequently, the scientific world is going towards total artificial heart. So that is the same pumping mechanism we might have seen in our English movies, that Iron Man. So they will have some kind of nuclear reactor inside our body which pumps the heart in that actor. So that is all possible in reality. Maybe another 10-15 years or even before that it can happen. Okay, so that's what the science is being advanced. And this is it's another differences are the innovation in the neurology domain. So here you see the X-ray of a skull, a person with some coiled wires are there. So there are certain conditions, your deep parts of your brain, if it get affected. In the earlier days, we just condemn the people. The people either they go for coma and they have to lose their life, or the life will be finished there itself. But in these conditions, still they can be made to live a better life, a meaningful life. So that is the advantage of this. Is called deep brain stimulator. So here they use the electrodes and they will make the non-functionalized brain region functional, because the brain is nothing but our electric circuits only there in the brain. So that's how the brain functions. It passes the current, asks if I want to close this finger or bend this thing, everything is commanded from here only through electrical signals only. So the same thing, if there is a deficit is there, we can substantiate, means we can substitute. So that's how this particular medical device is being utilized. Okay, so here the electronics engineer, electrical engineer, and material scientists, which they developed the compatible materials to this thing. So how those things will be done, that we'll see in this talk. So these are some of the major medical devices you can easily understand. So that I have given as example here. We'll go to the next level, that is the medical device. This is nothing but composition of all these interactions. As I said, truly interdisciplinary. So material, that is, it can be a metal, polymer, ceramic, or composites developed by a group of scientific people. Then design. So again, this comes from anyone. Even in this, I hope in the future some of you become an innovator and you will make new designs, medical devices in this area. I hope definitely it will happen. So that day you should remember me. So engineering and technology. So this is, anyone can conceive any idea and that can be materialized. So that is possible. Then comes the evaluation. So that is, today's my talk is around this area only. Physicochemical characterization, biological characterization, that is biocompatibility evaluation. So I will be focusing on this small area only. Okay, you see this is a very vast area. I am just touching upon the one small area, biological evaluation only. Then of course it will go to clinical use, where the clinicians, they will utilize depending upon the condition, and further they modify it, or they will again redesign it, and that's how the things happening. So that is the classical example is that mechanical heart valve, starting from open heart surgery to there is no surgery at all at this point of time. So that is the it has improvised. So that is mainly due to this thing. So everywhere, so the team, you have to open your things. You should not restrict yourself to one particular discipline, one particular science community, something like that. You should not be there in this genre. You have to open, you have to talk to any person, from accountancy, from avionics, from engineering, from medical, everyone. You can talk to them. Something will happen. Some interaction, some connection will happen. Some new things to the society will come in this thing. Okay. So open up your mind, open up your philosophies, principles, everything, and keep always hunting for new things. Okay. So we'll come to the main area. So as I said, the biomaterials, medical devices. Every world you will find lot of materials. This chair is made up of fabric, steel, plastic. So that is also non-living thing. Whether we can use it for some purpose, there is no. For biomaterials, there are certain characteristics are there, certain properties are there. So that's how the material differs from its routine material. Stainless steel, that is a classical example. This water bottle is made up of stainless steel, but this is not a biomaterial. Whatever this coronary stent which I shown, that is a stainless steel biomaterial. So what is that? So there is a composition. Alloys we might have studied in our elementary classes. In 6th, 7th we might have seen the alloys, different kind of alloys we studied. So there is a differences is there. Different SS, you have titanium, you have cobalt, chromium. So there are different materials are there. So they possess certain characteristics. So if you identify that characteristic, that can be utilized as a biomaterial. So that is possible only by evaluation. So in that, this biological evaluation helps us to identify those properties of a particular material. So that we will see. So we have metals, we have polymers. So polypropylene, polyethylene. So you can make polyethylene terephthalate as PET bottle, and the same polyethylene terephthalate fabric you can use a blood vessel. So only the form, the shape decides what's its function. Okay. So in that, so that is also only by evaluation you can understand this. So again, if you say biocompatibility, so there is a domain specific terminology called biocompatibility evaluation. So this kind of evaluating in the biological system is called biocompatibility evaluation. And the properties that particular material, either metal, polymer, or ceramics. Ceramics mainly used in the orthopedic area because they are the sturdiest material. So they are being used in the orthopedic region. Otherwise, the glass, what we see in our water glass, that is also a glass, ceramic only. So that is a object which we cannot use it for any medical purpose. But if you modify the composition, you can use it. Most of the dental filling materials, so teeth filling materials, is a glass, bioglass. They call it as the small anecdote they will give, bioglass. That means that can be used for biological application, that is medical application. So what that physicochemical property, if you understand, then you can use it. So that's how the biomaterial as well as the medical devices nothing but incorporate or use these specified or very specialized biomaterials and make the design changes. You, according to valve means it should be opening and closing, one way opening and closing, and if it is a coronary stent means it should be thin, it should be expandable, that is elastic, and it should be as tiny as a blood vessel. So such kind of design incorporation. So if that is there, then that is called a medical device. Okay, biomaterials as such and medical devices with design factors also. When the design factors, when we incorporate, how our body is going to accept or reject it, so that only through biological evaluation only we can understand. So that's where the biological evaluation of so-called biomaterials as well as medical devices helps us to innovate new medical devices as well as improvise the new biomaterials also. So that we'll see. There are again some of the things. So these biomaterials, when we place inside our body, it will change over a period of time. Either its surface chemistry, as we see, none of the substance in the world is stable. So called, we say everything is stable, but it is not so. As you are aware, you are better than anyone. So in the physics, we will see the neutron, proton, and electron, and there are different shells are there, and the electron jumps from low energy to high energy, or high energy to low energy. Something is there. So that is called this. Nothing in this is stable because it will change. Some materials it will take in a very slow manner. Some material it will take in a very fast manner. Okay, so that's all. So because of that elemental level interaction, this biological system also interacts. When you place inside any biological tissue, this so-called materials, it will interact with the surrounding and it will change. So that causes biological system to react. So that how it is reacting, what kind of reaction for different kinds of material, as I mentioned earlier, the metal, polymer, ceramic happening. If you are able to study that, understand that you can create new materials, or you can grade these materials or classify these materials, and you can find out which is useful, which is not useful to your application. That thing is. So that kind of initiate different tissue responses studying is called. So this we study in this biological evaluation. So this will help us to innovate newer materials. So this is the a small overarching pathway of any biological evaluation. So particular material is identified depending upon our application, as I mentioned earlier, in which domain you want to use it. For example, replacement of blood vessel or support of blood vessel. There is two terminology used. One is replacement, another one is supporting, that is the restoration. If there is a tubular structure is there, you just want to maintain the tubular structure. So where you use stent, where the tubular structure is completely gone. For example, accident, you lost one particular blood vessel. That means you have to go for a replacement. Okay. So there is this thing. So that is very. So depending upon your application, you have to choose a particular material. So for supporting, you need stent, metal. For replacement, you need polymer. Okay, artificial blood vessel. So that's all. So you have to select that thing. Of course, physicochemical characterization, you can make it. What is its physical strength, whether it's sturdy or not, whether it is pliable, flexible, all those things can be studied. Data's information available. Then it will come for biocompatibility evaluation. That is so called part of your biological evaluation. So where our body is made up of blood and tissues, cells and fluids, otherwise we call it as the major fluid is one is blood. So if the blood, if we get affected by the exposure to this material, we will end up with risk, that is loss of life. So naturally the hemocompatibility, how your material is friendly with the blood, so that has to be evaluated. So the terminology is called hemocompatibility. Then cytotoxicity, it is also, ma'am was telling about the wound healing product which they developed. The first one they evaluated is the cytotoxicity, how it is friendly with the cells, whether it supports the livability of the cell, that is being evaluated. So that is the first and foremost thing here in biological evaluation. So any biomaterial which is developed, first it will be tested for its cytocompatibility, whether it is supporting the living of any cells, because that is first and foremost thing, because our body is made up of completely cells. If it is going to affect the cell proliferation or even cell functionality, then that is useless. So that is the thing. So that is called. So that's why exclusively the cytotoxicity is being done as a preliminary development in biomaterial. Then the third one is toxicological evaluation. So toxicological evaluation means nothing but due to the presence of this so-called foreign substance, that is foreign material, whether any of our organ function, tissues function is getting affected. So that is being done. So that is called a toxicological evaluation. We will see some of the tests which is being done. So this is how the device development happens, and finally it will go for preclinical, that is animal trials. So it will be tested in simulated animal models. So this whole process is called biological evaluation of biomaterials, and what we try to understand from this is called risk assessment. So due to the presence of this so-called biomaterial inside our body, what kind of risk we have, what kind of benefit we will have. So it will be always weighed. If you have twofold benefit and there is one-fold risk, because these are non-living substances, so naturally we will have some amount of problem with that, whether we can manage it or not. For your understanding, I will tell you. I hope everyone have an understanding about blood's property. If you make a cut wound in your body, the blood will come out. How long it will come out? Anyone, can you give an answer? How much time the blood will come out from your body? A small cut. Or if the blood comes out of your body, what will happen to that? Clots. Okay, so that is the prime property of a blood. If somebody's blood is not clotting, there is a problem. Either the person has some disease, some disorder, something we might have studied in our earlier classes also, hemophilia. That is okay, non-coagulation of blood. So this blood clotting is very, very important, and not only blood clots, the science behind that is that blood, when it come in contact with foreign surface, the blood vessel is its native surface, and the blood vessel is lined by endothelium. That gives the property of non-coagulation, because its surface is made up with some receptors and some anticoagulant substances secreted by them. So that's why inside the blood vessel the blood is not clotting, it is just in the fluid form. If it comes outside the body, it will clots. That is called foreign surface when it comes. So there is intrinsic, extrinsic pathway. I hope I am touching about some of the things, somebody will have some idea about it, because this is also we studied from our biology plus 2 or 10th standard onwards. So intrinsic, extrinsic, that is tissue angioplastin, TPA they will call it as, and extrinsic means anything, glass surface or anything, if it comes it will clot. So that is also the same thing, just this is the background to understand this thing. So I told coronary stent, which is a metal, foreign substance. Okay, the surface is foreign substance to native blood vessel, which is inside your blood vessel, it is there. Whether blood will clot if it come in contact with that or not. Anyone? Metal is there inside the blood vessel. Whether blood will get clotted or not. Yes or no? Only give your answer. No? Anyone say yes? Anybody says yes? Blood vessel, inside that we are putting a metal. Whether this blood will get clotted or not. My question is clear, na? And I told you previously also, the blood's property is that when it come in contact with any foreign surface, it will clot. That is the nature of blood. Okay. So I given the clue also, I given the question also. Please answer me. Blood will get clotted. Okay. So that is a risk. But again, the science is there. A snake bites us. If a snake bites me, I will die. Why? Do you know any reason? Why snake bites? Blood clotting. Yes, there are two. again, that is also. There are certain snake bites which causes clotting of blood. So because of that, you will die. There are certain bites are there. Certain snakes venoms contain some anticoagulant factors, so that by losing blood you will die. Okay. So there are two kinds of that. So that substance which is not allowing the blood to clot is called a heparin. This you might have heard somewhere or studied somewhere. That is called a heparin. That is called an anticoagulant. So that is also being used medically for the benefit of human being. Okay, this is just ideas. What to use, where to use, when to use. So that's where a scientist is the inside. Every one of us, we have a scientific mindset is there. So but we are not using it. We are keeping him quite inside our body. So make use of him, that particular personality. So the same thing, if you keep a coronary stent inside the blood vessel, and if you give an anticoagulant factor, so we used to take these. I am not wishing every one of you. Some of you have a parent or somebody, your relatives. If they have a coronary stent inside the body, what tablets they are taking. You just ask them. Especially in the afternoon, they will take one tablet and they will say, this tablet will prevent my blood not to coagulate. And the name, commercial name is Cloplet. Okay, Cloplet is the tablet. Normally everyone will take this thing. So that is. So the risk associated with this biomaterial is clotting the blood. And that risk can be overcome by taking the anticoagulant. So that's how. So that's why I am telling this thing is, so this is again a scientific innovation. So any biomaterial will have a baseline of risk, but that can be managed by again some scientific tools. So that's how we are harvesting the benefit. As I told, the one-fold problem with the coronary stent is that clotting, but the two-fold is your life is saved. That is much better. So that's how if you take an anticoagulant, this can be overcome. So these and all through innovations will find it out. So there are now we have well established standards are available. That is called ISO 10993 Part 1 biological evaluation of medical devices. So they give the next slide. I will explain what and all it is explained in the thing. So using those standards, nowadays everything is structured. So we can understand biomaterials risk and how it can be managed and how the risk can be studied. There are methods are there that is also being explained. So if you understand this risk, we can understand the management. If you can assess the risk, you can manage this thing. And if you can do these two things, what you can benefit is, one is that foreseeable misuse of these medical devices and how can it can be overcome. So that is also can be planned. Then life cycle evaluation, in that also it is being used. Then post-market surveillance also, these medical devices when used in clinical population, what kind of complaints they are coming to the clinician again and why it is happened. Again, if you evaluate properly, you can address that conditions also. So all these things can be done through proper biological evaluation only. Okay. So this is the thing. So there are different kinds of medical devices. So surface contacting devices, externally communicating devices, and implantable devices. So what I shown, the mechanical valves, the coronary stents, all those things are called implantable devices. That is inside the a major organ, something it will be there. And depending upon the contact duration, that is the third column. So the risk can be calculated. And the last column, fourth column, that is the biological evaluation. So first and foremost one is the cytotoxicity evaluation, where your cell interaction with the material is initially studied, then sensitization, irritation, systemic toxicity, genotoxicity, hope many of you might have studied this one, implantation, hemocompatibility, how it interacts with the blood, that can be studied, and other toxicity. All these are called the toxicological evaluation. If you do these tests in animal models, you will be able to understand the property, that is so called the bio-friendly property of any biomaterial. And if there is a negative property is there, if we have some counter mechanism is there, we can manage that thing. If there is no such thing is there, naturally we have to eliminate or restrict the use of such biomaterial. So that's how the evaluation, that is biological evaluation, help us in developing a safe as well as useful medical devices. Okay. So hopefully most of these tests you might have heard somewhere else. There is chronic toxicity, carcinogenicity, reproductive and developmental toxicity, biodegradation, toxicokinetic and immunotoxicology. So these are standard toxicological tests used in the pharmaceutical areas also, as well as the same thing is being used in the medical devices also. The classical difference between the pharmacological agents and medical device toxicity is that pharmacological agents, for example tablets, syrups, whatever, if you take inside the body, it will get eliminated through your excretory system. Okay, so that is the thing. So that the risk is completely eliminated. That is not the case in case of medical devices. Once you implant something inside your body, it is going to reside in your body for as long as it is there. So that's why this risk has to be managed only, not eliminated. Okay, so that is one classical difference between classical. So here this is how the implantation studies through tissue material interaction, we find out the risk associated with any implantable material. The animal models mostly used to simulate the biological condition is small animals as well as large animals. So small animals they use rats, guinea pigs, rabbits, and large animals they use dogs, sheep, goats and pigs. And mainly the logic behind this thing is the longevity of these animals as well as your usage of your medical devices. In the earlier slide as I showed, certain medical devices we use it for few hours only, certain things for few months, And for example, valves and all things are, as long as the person is live, it has to be there. So depending upon that, what time, what kind of reaction will be there, we have to study. So that's how we choose small animals and large animals depending upon our usage of that particular material. For example, in goats, we can go up to 2 years, 3 years study, so that whether the material changes, property changes over the period of time, that can be understood. Okay, so here they use the animal models. They use one side, the same animal model itself, the test material which is newly developed or new innovations, and the other side is control animals which is existingly used, that is a well known. Okay, so how it is compared, whether better, inferior. So based on that, they will take a decision. Okay, this is a useful biomaterial or medical device, or it is a not useful. Again, you have to improvise it. So that's how this is. So there are in the histopathology, that is in the pathology domain, they use certain techniques that is called the histotechniques. So where they make from these tissues microscopic glass slides, and these microscopic glass slides will be evaluated using microscope, so that the cellular level changes. What is happening at the cellular level, tissue level changes can be studied well. Based on that, you make the decision. That is the report, that is called whether good or bad. So here, that is the cycle. So the final one, that is glass slide for microscopy, is the specimen which we evaluate in the pathology laboratory. So this is about the soft tissue evaluation is concerned. So there is certain improvised or modified techniques also there. So there is hard tissue. For example, if it is bone, which is very hard structure, which cannot be cut using ordinary microtomes. So again, these are very domain specific things. Maybe I hope you will get some exposure somewhere in pathology laboratory where you can find out why it is being used, what is the purpose, everything done. So just you have few pictures in your mind, so that in your future, if you come across any such thing, you will understand better. So here, the final one, that is microscopy. There is a bone, yellow color with a black color substance, is nothing but a metal, and the bone. It is a microscopic picture of a bone interface. So that also can be studied using histotechnics. Okay, so this is again a muscle. So here, as I said earlier, that small animal model I showed now. So in that, in a muscle, they will put this kind of small metal rods, and they will evaluate the interface under microscopy. And in a magnified microscopy image, it will appear like this, the implant site. At 4 weeks, you will find a wound with inflammation. At 12 weeks, you won't find a wound at all. You will find only a scar tissue. If this is a response to a biomaterial or medical device, they call it as a safe medical device. So as simple as that, if any wound, if it heals, it is good. If it is not healing, there is something problem is there. That means the material is changing its property. So that cannot be used as a safe device. So that is the simple logic behind this biological evaluation is concerned. Okay. So as I said, this is the valve. So the other side, two Chitra TTK valves. So that is the indigenously developed mechanical heart valve from the Sri Chitra Institute. So this is equivalent to similar the tilting disc model, and this is the valve placed inside a heart of a preclinical animal model. Okay, so you can see this is from the auricle view from the atrium. This is how inside the heart this will be there, and the white color is the disc. It will open and close so that the blood flow will be unilateral. And the bottom one is the from the ventricle side we are viewing the valve. So that is the disc, and you can see. So this is the thing. So here what they will see is that when the presence of this material, healing response, again it will causes the obstruction of the flow area or not. So that is the major thing. That is a challenge, this thing. So they make such a material or they make such a design so that the tissue ingrowth won't be there. So that only can be confirmed using histotechnics only. So through microscopic evaluation, the the annulus white color area, the surrounding white color area will be a small piece will be taken, and histology using histotechnics we make the microscopic slide, and you see it under microscope, and what kind of response, healing response is there or inflammatory response is there, that will be studied, and based on that they will take a decision. Okay, this is a very good material for your valve cage or valve sewing ring, something. So that's how evaluation helps. Then as I said, the stents. So here also they will place these stents inside the blood vessel, and after that, so this is the again a preclinical animal model, heart with, so the small 2 rounds, that is the X-ray image of a heart. Okay, big heart X-ray, and inside the blood vessel you have the stent is there, and the next one, bottom one is the inside the cut open. This is the diameter of this blood vessel is 2 millimeter. Okay, these are somehow magnified images, and you can see the shiny metal struts inside the blood vessel. Okay, so this also again not only this is sufficient, you have to see it under evaluate using histotechniques also, that is microscopic evaluation. So this is nothing but the microscopic evaluation of same specimens. Blood vessel, the small black studs are nothing but the cross sections of the metal studs. So this can be evaluated so that what is the tissue response, whether it is aggravated or not aggravated, those skin can be studied. Okay, so and this is the third example which I showed, that is the total replacement of the blood vessel itself. You can see the top one, so there is a heart and the major blood vessel, that is the aorta. So there is a major blood channel through which the whole body is getting the blood. If there is a disease, there are certain disease conditions are there, so in that you have to replace the whole aorta. So this is same thing, the polyethylene terephthalate, which aside fabric made vessel, blood vessel, so that is being evaluated here. So mainly here they will see both the tissue response as well as the blood non-blood clotting ability. So here, as I said, along with this material, the person will be maintained in the anticoagulant. The blood will contain the anticoagulant and preset quantified level. So this is the histology. So how much the tissue response is there, whether healing response is there or inflammatory response is there. So on that, this is a continuous process, so that will be evaluated. So this is how they make the decision. At the junction, if there is no blood clot and the tissue healing is perfect, they say it is safe. If it is not so, a continuous ulcers or blood clots, if something is there, they say it is not a no. So here only we evaluated safe animals, so that's why I am just putting the safe histology pictures. So that's about. So as a conclusion, so the medical devices are biomaterials we use for replacement of any diseased organs or tissues, sometimes for restoration. As I said, the coronary stent, we can use it as a restoration just to maintain the tubularity, or regeneration. That is the recent advancement in this area is, if any organ, a small tissue is dead or deceased, you can totally replace. So that is called, initially it is called tissue engineering. Now the new domain name is bioprinting. That is a whole organ. You might have maybe 10 years back itself, you might have seen a good picture of a rat with our ear on the back of a rat, that come in most of our daily newspapers. And even they have now kidneys, liver, everything. There are advanced stages, it has come into advanced stages. So those organs can be printed, means outside your body you can create these organs and you can put it back inside the body. So that is the so-called regeneration in this medical device domain. So everywhere this evaluation is very, very paramount. From the beginning stage, what materials you have selected, what kind of design you are going to incorporate, and where purpose you are going to use. So all these things can be evaluated using biological system, either in the animal models or in the in vitro system. So it is a critical process and it is a continuous process also. That's how the biological evaluation helps in the innovation in developing new biomaterials, medical devices. So with this, I am just closing my talk, and if you have any pressing questions, basic questions, you can ask me.
Thank you very much for the informative talk. Now the session is open for discussion. So we have time for 1 or 2 questions from audience.
Thank you, sir. Yeah, I would like to know about what are the different techniques in your institute you are using for the distribution studies of some of the materials like nanoparticles. Is there any criteria or study techniques are following there in your institute?
Nanomaterials.
Yes.
So nanomaterials, mostly they will use it for any as such as a physical form. Whatever I have shown here is mostly the physical form materials, support as a restorative or replacement purpose. Nanomaterials mainly they use for delivery of certain items as far as therapeutic aspect is concerned. And diagnostic aspect, that is totally different. I am not dealing with that this thing. Therapeutically they will use drug delivery systems, such kind of things. So again, these are nothing but synthetic materials only. So the same kind of evaluation, so the toxicological evaluation from sensitivity to acute toxicity, the same battery of tests only being done for the evaluation of nanomaterials when it is recommended for therapeutic use. So the same kind of toxicological evaluations.
Yeah, I mean the distribution, because how it is localized in different organs like that.
Yeah, so all those things that is covered in the toxicological evaluation.
Toxicological. Okay.
Yes.
Thank you, sir.
The first tech.
Sir, thank you very much for the wonderful presentation. I have a question. What about the stability of the stents? How long it will go for in an individual? And if it is not stable, what happened? What will happen to the stent material?
Yeah. So as far as I said, the physicochemical characterization. So that is the first and foremost thing. When you say application, for this particular application, we are going to use this thing. So these materials, our normal lifespan is somewhere around, say average, say 70 years. And these materials so far tested, used in the coronary stents, the benchmark itself 140 years. As long as this much time, that material has to be stable in that form. As I said earlier itself, there are some materials slowly they will degrade, so called our half-life. Okay, so based on that already they have benchmarked certain things. So whatever the sturdy non-degradable metal stents which so far developed, they will be there in that condition in the physical form minimum 100 years. So that's what the benchmark they already set. Based on that only, the same thing if you say knee replacement, titanium based things which are being used, the same thing, they are also as long as 100 up to minimum 100 years they will be in that form only. There won't be anything unless otherwise there was a articulation associated corrosions, so that is inevitably some other thing. So based on, so that's all. And based on these experiences, now the stent has moved to next generation called the degradable stents. Okay, so in that case there are certain materials which will, without any harm, they will degrade in our body. So that is the newer innovation in the coronary stent is concerned. So that's what it is. Every 10 years there are many breakthroughs are happening in this particular medical device is concerned. So that's why from replacement to restoration, now the present concept of all biomedical innovation is towards regeneration. Because as long as this foreign substance is inside your body, you have to take this anticoagulant, for example, in case of stent, that is a risk. So if it is not there, only it serves the purpose. And any disease condition is a temporary thing only. Okay, all of us will get disease, after a week will get cured of fever. Same thing with the other tissues, organs also. There is a temporary thing only. So that much time if you support, naturally our body will heal it. So that is the principle behind the so-called regenerative medicine. So that's how the 3D bioprinting, tissue engineering, all those comes. The present lifeline of all our human beings. So that's all. Thank you.
The first technical session is over. Now may I invite Dr. Sandhya to give the token of appreciation to Dr. Sabarishwaran.
Thank you, ma'am. We shall now proceed to technical section 2. I invite the section chair, Dr. Linda Lewis, Associate Professor, St. Xavier College, Aloha, to introduce a distinguished speaker, Dr. Sibin Tia, Professor in the Department of Biochemistry at Sri Chitra Tirunal Institute of Medicinal Science and Technology, as he takes charge of the upcoming section.
Good afternoon. Actually, as we know, biochemistry is the mother of all the biological sciences, and it is having roles in almost every field like molecular biology, biochemical genetics, protein chemistry, and so on. And we have an eminent person amongst us, who is doing research in all these areas. When we look at his profile, his scientific profile, it is really fascinating, and it is my great pride and privilege to introduce our next speaker, Dr. Sibin T. Raghavan. And actually, sir is currently working with advanced proteins. They are actually conjugated proteins as far as my knowledge, and they are having effect in various disease formations, and that's why his topic is today is dealing with the amino acid disorders and developmental disorders. And when we look into his research profile, his scientific profile, he is into research for last more than 25 years, and of these almost half a decade of international experience as postdoctoral fellow. And he is currently having projects on cardiac fibrosis as well as on Parkinson's disease, and it is our great opportunity to listen to him, a person with number of research publications in high impact international journals. Again, he was a part of developing the research tools or the identification tools for COVID diagnosis, as well as he is having papers on molecular biology on various methods of DNA isolations. So while I looked into his profile with great enthusiasm, I'm inviting you, sir, for this talk. Thank you so much.
Thank you, ma'am, for enlightening us. Now I invite Dr. Sibintia to handle the technical section.
First of all, thank you, madam, for the kind introduction. And let me congratulate the team here for organizing such a wonderful seminar, national seminar, on a very short notice. And a special congratulations to Sandhya ma'am on her retirement and having a happy and prosperous life after retirement too. So I will try to make this as short as possible, because I think everybody is waiting for their lunch now. So almost two talks are very difficult. One before the lunch to keep you away from the food, and the second thing is after the lunch to keep you awake. So so my talk today is on amino acids and developmental disorders. I'll try to make it a bit short. I just wanted to have this slide just to show my career path for the students. So I started off with doing my basic biochemistry graduation in Madras University. So at that time, my only aim was to do my PG in biochemistry. So when I went for my PG, I had one professor who said, when you come after your degree, you think that you have learned something. But when you come for an MSc, you will know that you have just learned a bit, you have a lot more to learn. But at that time, that professor was a PhD, and he said that one word that the whole class laughed into. What he said was, after BSc you will say that you learned a lot. After MSc you will learn that you have learned only a bit. And to quote my professor, he said, after PhD you will learn that many people around you know only very little about the subject. So it is a vast field. It is education, or whatever degree you're doing is just a knowledge, enhancing your knowledge. Every day is a learning experience. So this is for the students. Try to learn as much as you can, just not for degrees, but to earn your knowledge. A little bit about the institute. Dr. Shabarishwaran sir has given you excellent view on the device part. So Sri Chitra Tirunal Institute is an institute of national importance at Trivandrum. It has three wings, in fact: the hospital wing, the biomedical technology wing, and the Achyuta Menon Center for Health Science. So hospital wing, it is a tertiary care referral hospital for neurology and cardiac associated diseases. Basically, they have research education and the patient care in the hospital wing. So they cater to, apart from the patients of cardiac and advanced cardiac and neuro disorders, they have courses like post-MD courses in neurology and cardiac specialization. And of course, there is a PhD program in the biological sciences. So again, biomedical technology wing, as sir said, they are into developing devices, and they also run a PhD program and master's program in technology. Achyuthamenon Center for Health Science Studies is again a unique of one of its kind in the country, which makes policies for the healthcare. So they are one of the pioneers in making healthcare policies. They do have MPH and PhD programs. So just have in your mind, this is a good place to do your PhD studies for the MSc students. Okay, so my talk today is on amino acids and development disorders. So as you know, the students, biochemistry students, you are here, right? Still awake? Okay, so biochemistry students would have been, would have learned the basics in biochemistry first. They would have learned about carbohydrates, amino acids, proteins. So the first class they learn is amino acids are building blocks for proteins, right? Amino acids are building blocks for proteins. But I don't, in this talk, I'm not going to concentrate on that particular aspect, but how the levels of amino acids in our plasma can be correlated to some of the neurological disorders. That is what I'm trying to make. So neurological disorders, you have two types. What I am focusing on is on two types. On the left-hand panel, you have the neurodevelopment disorders like the intellectual disability. You have the autism spectrum disorders, and then you have global development disorders. Global development disorders specify that there are more than two disorders together, so that is why it's called global development disorders. And apart from that, there are other childhood onset, all related to the brain, but the development happens at birth or at the early stages of life. But the other childhood disorders could happen later on during childhood, not at birth or not at the early childhood also. There are disorders like genetic disorders which can happen after 10 years or 12 years. Those could be genetic, but they arise not at birth but after a certain period of life. So the other childhood onset disorders include inborn errors of metabolism. Inborn errors of metabolism are quite often thought to be some neurological disorders. Only they come to a neurology clinic, only after a series of tests they come to know that it is not a neurological disorder, but they have some deficiency or inborn errors of metabolism. Those are not actually classified under the neurological development, neurodevelopment disorders. And then you have neurodystrophy and movement disorders. These are some of the aspects. So why amino acids? Why amino acid profiling is important? As I said, amino acids are fundamentals to brain development. They act as neurotransmitters, like glutamate and glycine are neurotransmitters. And some of the amino acids like tryptophan and tyrosine are precursors for neurotransmitters. They are energy substrates and signaling molecules. Now, the objective biochemical evidence in children have shown that amino acid profiling can be seen in patients with global development disorders, intellectual disability, autism spectrum disorders, and other related diseases. So plasma or serum amino acid analysis is now becoming more and more relevant at recent times for diagnosis. Earlier, when a patient used to come for a neurological disorder, in general, neurological disorders or development or something related to neurology, there is no specific blood marker or there is no diagnostic marker to say that this marker can be linked to this particular disease. So mostly these are done using imaging tools or their behavioral changes, their activity, all those are checked to come to conclusion that this particular child or the patient is having a particular disorder. But blood and other parameters are rarely used. But more recently, there are certain markers neurologists also prefer to profile. So using of plasma amino acid profile can be used to differentiate between the neurological disorders, as well as it can be differentiated as inborn errors of metabolism can be differentiated using a blood parameters. So again, there are emerging evidence to show that children with autism spectrum disorders, intellectual disability, exhibit altered excitatory and inhibitory balance. So there is something called glutamate GABA axis. Glutamate is a excitatory neurotransmitter, and GABA is an inhibitory neurotransmitter. So you need to have a balance of this to have a proper cognition. So once this balance is lost, it might lead to some neurological disorder. Apart from that, disturbed tryptophan metabolism. Kynurenine pathway is something, but it's a degradation of tryptophan. That pathway is very important because I'll come to that. That is because there are certain intermediates which are neuroprotective, and there are certain intermediates which are not good for the neurons. These patterns may reflect mitochondrial dysfunction. That means the amino acid patterns may reflect mitochondrial dysfunction, indicate oxidative stress and neuroinflammation, and serve as a disease stratification agent. So I was talking about kynurenine pathway. This is from the biochemistry textbook, the kynurenine pathway. It is all about conversion of tryptophan. Tryptophan, initially it is converted to N-formyl L-tryptophan, but in the series of steps it is converted to various intermediates. There are certain enzymes which are very important here. One is the first enzyme is called the IDO, or indoleamine dioxygenase, which converts tryptophan to N-formyl tryptophan, which in turn is, with the help of kynurenane's formamidase, is converted to L-kynurenine. Now kynurenine is present in many parts of our body as kynurenine itself, or it can be converted to 3-hydroxykynurenine, which is highly reactive. 3-hydroxykynurenine is not stable but highly reactive, and by the series of enzymes. So all these enzymes, kynurenine formamidase, kynurenine monooxidase, are important because now kynurenine, if it is converted to kynurenate, it is a neuroprotective agent. But if the kynurenine is converted to 3-hydroxykynurenine, which is highly reactive, or which in turn is converted to 3-hydroxyanthranate or quinolinate, are neurotoxic. So the same pathway, you have intermediates which are neuroprotective or neurotoxic. So all these, taking all this, we planned a small clinical study or retrospective study with the patient data we had. This is not an experiment we planned. We didn't have any objective out of it. So we had some data in the hospital, and we thought how to make use of this. So this is a retrospective study to characterize the amino acid alterations in pediatric neurodevelopment disorders and evaluate diagnostic susceptibility using a multivariate analysis. So this is the study. The design was very simple. We had 300 patients, 303 children in the age group of 2 to 17 years, and the data was collected over a period of 5 years from 2017 to 2022, and it was a male-dominated population. So moving on to the study design as such, as I said, it is a male-dominated. It is we had more male than female in the study. There are 195 in number. And if you see the right-hand panel, it is the distribution of subject across the diagnostic groups, and it is kind of scattered. Like you have more patients with ASD or autism spectrum disorders and global development disorders, but others are more or less the same. So it is always not, when you plan for an animal experiment or when you plan for a small experiment, it is always easy to make an analysis. You use this particular tool and you do your statistics. But when you go for a clinical study, when there is a lot of variations in the sample, the kind of people there, different diagnostic groups, the analysis becomes a bit different. So we contacted a biostatistician to give us a good tool for diagnosing these things. This was the machine we used, the amino acid analyzer from Hitachi, and it could measure amino acids as well as some of the derivatives of amino acids. So you might be wondering, there are only 20 amino acids and you have listed a big list with more than 20, but this includes the 20 amino acids and the derivatives from amino acids. This is how the peaks look for the standards, and we can compare the peaks of our sample and the standards, and we can quantify the levels of each amino acid in the sample we have. So basically it is just plasma. Plasma is used. There is no purification, nothing is required. Plasma as such can be injected into this machine. There is a pre method which happens within the system. So there is no preparation as such. The plasma can be injected into the system, and you get peaks like this which can be quantified. So moving on to the results, again I will try to make it short. Altered levels of alanine and glycine, these are amino acids, across different diagnostic groups. You can see there are 22% there is of global development disorder, there is an alteration. Now when I say alteration, it could be either increased or decreased. Being a patient population, there are certain cases we have fixed a normal range for these amino acids. It could be higher or lower. So, some of the results I have represented as higher or lower or altered, and which is higher I have written as higher. So alanine and glycine are frequently altered in global development disorders as well as in epilepsy. Now this is again gamma aminobutyric acid and taurine levels, and you can see there is a significant GABA is significantly reduced across all diagnostic groups, whereas taurine was significantly elevated in certain groups like ASD or global development disorders and epilepsy. The altered levels of another derivative to amino adipic acid across diagnostic group was checked, and we found that again there was a significant alteration in these 2 amino adipic acid levels. 2 amino adipic acid levels significantly was elevated in global development disorders and movement disorders, and the levels are given on the right hand panel. Don't worry about all the colors there. It is a multivariate analysis wherein the variability factor was taken into account. The variable importance in projections called VIP score. So the higher the VIP score, the greater the contribution towards the contribution. So on the right hand panel you can see the levels of these particular high or low as marked in red. Red means significantly higher, blue means significantly lower, and all the boxes there have shown that different diagnostic group how the particular amino acids has framed or derivative has framed. So ASD and GDD showed the moderate but consistent shift in this score. This is again a principal component analysis wherein the 4 different components were analyzed, and this is just to show you that the multivariate analysis of amino acids and disease classification showed significant overlap across diagnostic groups, indicating that amino acid profiling alone cannot be used as a diagnostic marker. So you might be thinking all this work has gone waste. No, this is just a finding, just to show that with available data we were trying to make a predictive if one particular diagnostic amino acid can be used as a diagnostic marker, but we were not able to specifically point out that this particular can be used as a predictor. It can be used as predictor, but it cannot be used as a marker alone. Now this study is continuing. In this study we had only 300 samples. Now we are trying to expand it to a higher number so that we can have a better correlation. So at this stage, this is what we have. This is a summary of the work I presented. There were 303 pediatric patients or pediatric neurodevelopment disorder cohort, ranging from 2 to 17 years of age. Their fasting plasma was used for amino acid profiling using an amino acid analyzer. And the major findings were there was a significant decrease in the regulatory, significant decrease in the inhibitory neurotransmitter GABA, and there was a significant increase in the neuromodulator taurine. And there was an inhibitory excitatory imbalance in neurological patients there was metabolic overlap between various diagnostic groups, but there was no disorder-specific clustering found. So, amino acid profiling reflects shared metabolic dysregulation, useful for screening, but cannot be used as a standalone diagnostic marker. So, with this, I will end my talk. I would like to acknowledge all these people because most of the work was not funded, but done using the sample given by patients. Okay, so the Dr. Soumya is a neurologist who identifies these disorders, and if required, she sends us samples. Dr. Deepa, Srikala, and Divya were the people who have actually performed this amino acid profiling. Dr. or Lakshmi was the person who has analyzed most of this multivariate analysis and things. And Dr. Srinivas and Dr. Madhu are my colleagues in the department. And special thanks to all the participants in the program, because getting samples itself from pediatric patients is difficult, and we had a good social work team who they convinced them so that we could get some samples for this particular study. Thank you, and I'll be happy to take some questions if any.
Dear participants, now this session is open for discussion and you can have your queries.
Dr. Sibyl, I would like to know about you have checked the different disease condition of children's, the samples from different children having different disease conditions.
Yeah.
And is there any particular amino acid present in any of the disease? I mean the epilepsy or ASD or any disease conditions? Any particular amino acid dominated or present?
Yeah, so it is that is what I am trying to say. We cannot say that this particular amino acid is elevated in this condition or it is reduced. It is a mixed population. So further studies are required to come into conclusion that whether a particular amino acids can be referred to for this particular disease. That point we have not reached.
Okay. Thank you.
Sir, actually I have a very innocent query.
That's okay.
Why? Because this is for the first time actually I'm listening for someone who is analyzing amino acids for such disease disorders. What I had the query is whether this amino acid content in the serum will vary with our food.
It could vary, but that food parameter, this is a very good question. We have not checked that aspect. We have not taken diet into aspect because we cannot, in this age group, we cannot ask them to follow a specific diet.
Okay.
And that was not part of our study. As I said, it was a retrospective study with whatever data we had. Now in the future, when we plan some study like that, we can ask them to take a specific diet and then check this. But diet definitely would matter in this.
Okay, and again one more thing, that along with the standard amino acids you have shown us the derivatives including our neurotransmitters. So actually we used to go for RIA or such techniques to identify this neurotransmitters and all. And whether this analyzer is that capable to identify that very low concentrations?
Yes.
Okay.
So it is, it's a Japanese technology. So it is sensitive enough to nanomolar scale. I am not sure whether picomolar scale is possible. For nanomolar scale it is possible.
And one more query for the researchers over here, whether the instrumentations of Sri Chitrathirunal Institute is open for the students to access.
That is a very tricky question, which it is more or less accessible. You have to contact the particular person where the instrument is. It is more or less accessible. It depends on the availability because some of the instruments are extensively used. So you might have to book your slots, like you will ask them when is the best time to come. But more or less, most of the instruments are accessible to everybody. Yeah.
So can this be used for prenatal diagnosis?
I'm not sure. Maybe it can be used.
Any more queries? So with this we are coming to end of the session. So now I request our dear Dr. Sandhya ma'am to give away the token of gratitude to Dr. Sebin. Sebin sir, thank you for this wonderful session.
We will now pause for the lunch break. The rest of the seminar proceeding will resume at 1:55 p.m. We request all participants to kindly reassemble on time for the technical section of Dr. Sandhya. In parallel to technical section, poster presentation will be held at room number 203 and paper presentation at IQEC Hall, room number 207. Lunch arrangements are made in Xavier Hall. All participants are requested to reassemble on time. Thank you.
ലൈവ് ആണോ?
ഫോട്ടോ രതീഷ ഓടി വാ. രതീഷ് ഇവിടെ വന്ന് നിൽക്കുന്നില്ല.
കാണുന്നുണ്ട്. സൈഡ് നോക്കാം. അതെ അതെ ആയിട്ടില്ലോ, ക്ലിയർ ആയിട്ടില്ല. വരുന്നുണ്ട്, പക്ഷെ അത് സൈഡ് കാണുന്നില്ല. അത് അതിനകത്ത് ഷെയർ ചെയ്യാമോ? ഷെയർ ചെയ്യാമോ?
എന്ത് ഇതല്ലേ അനക്കരുതെന്നൊക്കെ പറഞ്ഞത്. ചോദിച്ച നീ അറിയാൻ നിന്റെ ബാഗിൽ ആരെങ്കിലും അതാണോ അനക്ക് ഇതല്ലേ അനക്കരുതെന്ന് ഇത് അനക്കാതെ വെക്കാനും പറ്റത്തില്ല. ലൈവ് അവരെയും പറഞ്ഞു അവിടെ മുൻപീന്ന് പറഞ്ഞിട്ട് ലൈവിൻ്റെ നിൽക്കുന്നത്. ആരും കാണില്ല. ഒരു കാര്യം ചെയ്യാം. ഒരു കാര്യം ചെയ്യാം. നീ പിടിച്ച ഞാൻ അങ്ങനെ പൊക്കാൻ പറ്റത്തില്ല. ഈ ചാർജ് ഇവിടെ സ്ട്രെയിനാണ്. വലിയ വലിയ കുറച്ചുകൂടെ ഫ്രണ്ടിലോട്ട് കയറ്റി വെക്കണം. പെരുന്നച്ച് അടിയിലേക്ക് വെക്കാം. അടിയിലേക്ക് അത്ര അടിയിലേക്കില്ല. എൻ്റെ ഈ ചാർജ് നല്ല പ്രശ്നം. എൻ്റെ പത്ത് കാലം ഇങ്ങനെ കയറി ഇല്ല പതുക്കെ കേട്ടോ. ഇനി അനക്കാതെ അങ്ങ് പോകുന്നേ മക്കള്. അതുപോലെ ഇത് ഓഫല്ലേ? ഇത് ഓണാക്കിക്കാൻ അപ്പുറത്തല്ലേ വെച്ച് അല്ല ചുമല കത്തിയോ? ഇവിടെ ചുമല കത്തിയതല്ലേ? കത്തി ചാർജായ എന്നുവെച്ചാൽ ചാർജ് യുവർ ഡിവൈസ് എന്നാണ് പറഞ്ഞേക്കുന്നത്.
വലിയ താമസമില്ലാത്ത തുടങ്ങും ആശംസ വരുന്ന കമ്മ്യൂണിക്കേറ്റ് ചെയ്യാം പിള്ളേരെ എത്തിക്കാറില്ല ഞാൻ കുറച്ച് പേര് ഇങ്ങോട്ട് വന്നോളാം ഹസ്ബൻഡ് ഓഫീസുണ്ട് ആശംസ ഉണ്ട് സെഷൻ ആണോ നടക്കുന്നത് ചെയ്തു തുടങ്ങുന്നത് അപ്പോൾ പിള്ളേരെ എല്ലാം കേട്ടിട്ടുള്ള കാര്യം പന്നസ് കഴിച്ചോ കഴിച്ചോ പക്ഷെ മഞ്ഞും ഇനി ഒരു സെഷനോടെ കഴിയാനില്ലേ ഉള്ളൂ ഞാൻ മഞ്ഞുസ് എന്നെ വിളിച്ചു അപ്പം 4 സെഷനല്ലായിരുന്നു സമയം ചോദിക്കുന്ന പക്ഷേ രണ്ട് സാധനം എളുപ്പം നടക്കുന്നുണ്ടല്ലോ ഞാൻ ശരിക്കും ഇവിടെ ഓടി വരിക ഞാൻ പെട്ടെന്ന് കഴിച്ചിട്ട് വന്നതേ ഉള്ളൂ ഞാൻ എളുപ്പം കഴിച്ചിട്ട് പിള്ളേരെ കാണുന്നില്ല എല്ലാവരും കഴിച്ചിട്ട് വരുന്നുണ്ടായിരുന്നു ഞാൻ അച്ഛനെ വിളിക്കാം പോയി ഇന്നത്തെത്തില്ല ഇവിടെ അച്ഛനെ ഇടയ്ക്ക് ഞാൻ പോകാനില്ല അച്ഛൻ കൂടെ ലക്ഷ്യം കാണിക്കില്ലേ നിങ്ങൾ വരുന്നുണ്ടോ ഞാനല്ല പക്ഷെ എല്ലാവരും രണ്ട് കിലോയിലായിരുന്നു അതൊക്കെ എന്നാണ് എല്ലാവരും നമുക്ക് ഒരുമിച്ച് പോകാൻ
എനിക്കറിയത്തില്ല ഇതില്ലായിരുന്നു എല്ലാം കഴിഞ്ഞു ഈ കാര്യമൊക്കെ ആയിട്ട് കഴിഞ്ഞു ഈ സാധനം ഓൺലൈനാണ് ഇതാണ്